hidden within the human genome. Among the 20,000 genes that serve as building blocks of life are flecks of DNA left behind by viruses that infected primate ancestors tens of millions of years ago. These ancient hitchhikers, known as endogenous retroviruses, were long considered inert or junk DNA, defanged of any ability to do damage. In a study recently published in the journal Science Advances, researchers revealed that when endogenous viruses are reawakened, they can play a critical role in helping cancer survive and thrive. The study also suggests that silencing certain endogenous retroviruses can improve the efficacy of cancer treatments. This study shows that diseases today can be significantly influenced by these ancient viral infections that until recently very few researchers were paying attention to. Research shows approximately 8% of the human genome is made up of endogenous retroviruses that slipped into the cells of our evolutionary ancestors, coaxing their hosts to copy and carry their genetic material. Over time, they infiltrated sperm, eggs, and embryos, baking their DNA like a fossil record into generations to come and shaping evolution along the way. Although they can no longer produce functional viruses, research has shown that endogenous retroviruses can act as switches that turn on nearby genes. Some have contributed to the development of the placenta, a critical milestone in human evolution, as well as our immune response to modern-day viruses like COVID. There's been a lot of work showing these endogenous retroviruses can be domesticated for our benefit, but not a lot showing how they might hurt us. To explore their role in cancer, researchers analyzed genomic data from 21 human cancer types from publicly available datasets. They found that a specific lineage of endogenous retrovirus known as LTR10, which infected some primates about 30 million years ago, showed surprisingly high levels of activity in several types of cancer, including lung and colon cancer. Further analysis of tumors from dozens of colorectal cancer patients revealed that LTR10 was active in about a third of them. When the team used the CRISPR gene editing tool to snip out or silence sequences where it was present, they discovered that critical genes known to promote cancer development and growth also went dark. Experiments in mice yielded similar results. When an LTR10 switch was removed from tumor cells, key cancer-promoting genes, including one called XRCC4, switched off too, and treatments to shrink tumors worked better. Cancer cells express a lot of genes that are not supposed to be on, but no one really knows what is turning them on. It turns out many of the switches turning them on are derived from these ancient viruses. Notably, the endogenous retrovirus they studied appears to switch on genes in what is known as the MAP kinase pathway, a famed cellular pathway that is adversely rewired in many cancers. Existing drugs, known as MAP kinase inhibitors, likely work, in part, by disabling the endogenous retrovirus switch, the study suggests. The authors note that just this one family of retrovirus regulates as many as 70 cancer-associated genes in this pathway. Different lineages likely influence different pathways that promote different cancers. People age, their genomic defenses break down, enabling ancient viruses to reawaken and contribute to other health problems too. The origins of how diseases manifest themselves in the cell have always been a mystery. Endogenous retroviruses are not the whole story, but they could be a big part of it. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.